Hello everyone, I am Dr. Amardev Rajesh Vatapati and I am a qualified bioinformatician. I did my masters in bioinformatics from SWIMS University and PhD from Acharya Nagarjuna University, Guntur. Today I would like to present the need and importance of my thesis work and the structure of my thesis work. The title of my thesis is Molecular Biocontrol Studies through active site cloning, in silico characterization, and structural analysis of juvenile hormone epoxide hydrolase gene of Helicoverpa armigera. Coming to the importance of my work, the Helicoverpa armigera ranks top among the agriculture insect pests in the world due to its polyphagous nature, elevated reproductive ability, and for its distribution worldwide, which includes Asia, Africa, and some parts of Europe. Helicoverpa armigera infests several economically vital crops like cotton and a variety of food crops like vegetable crops, legumes, and other important crops. It is reported that a multi variety of crops were infested by Helicoverpa armigera along with the cotton crop, resulting in great damage to Indian economy. To control the infection, latest strategies were practiced to restrict the insect development rate. Insect control strategies like area wide management, which is called as AWM, and integrated pest management, which is called as IPM, were practiced to resist the insect growth. But control strategies needs the combined efforts of the study of biology of the insect and culture of the insect, as well as the existing chemical insecticide options, whether it is a chemical pesticide or a biopesticide like NPP and BT. Controlling the damage had become a challenge to sustain the agriculture economy due to their low potentiality. Hence, exploring eco-friendly strategies had become the only option to control the Helicoverpa population. Controlling lepidopteran insect population by altering its own system using insect's own engine is a promising strategy that is focused much as damaging is specific to the target and eco-friendly. Studying juvenile hormone pathway as pest control strategy is considered to be the best control the lepidopteran insect population because juvenile hormone regulates the insect physiology, growth, and reproduction. A decade-old research on juvenile hormone pathway states that blocking JH metabolic pathway results in immediate death in the early instar stages of the larvae of the insect. Juvenile hormone epoxy hydrolase, which is called as JHEH in short form, is a hydrolytic enzyme from alpha B hydrolase family, and this JHEH damages the juvenile hormone, which is called as JH, irreversibly. Understanding the JHEH and its role in JH metabolic pathway is considered to be more overriding in view of the fact that very limited information on JHEH is available as of now. To implement chemical-free insecticide control strategies, understanding the life cycle and biology of Helicoverpa armigera is much important, which will be much helpful in designing the insect specific and eco-friendly third generation insecticides. Repeating my title, Molecular Biocontrol Studies through active site cloning, in silico characterization, and structure analysis of juvenile hormone epoxy hydrolase gene of Helicoverpa. The content of the thesis is organized into 7 chapters. Chapter 1 presents the introduction in relation to the title of the thesis along with the key concepts of the topics like damage to agriculture industry by the insect, relevance to the Helicoverpa, existing pest control strategies, control, a role of juvenile hormone and JH metabolic pathway in insect pest control, JH degradation mechanism, juvenile hormone binding proteins, which are called JHBP, role in transporting the juvenile hormone, along with the outline to various parameters carried out in the thesis. Chapter 2 is covered with the details of review of the literature on Helicoverpa armigera and its importance on agriculture industry, scientific taxonomy of Helicoverpa armigera, life cycle of the insect, mode of damage by Helicoverpa armigera and its importance on economy, review on importance of juvenile hormone and JH pathway, and mechanism of JH degradation, and mechanism of catalytic action of juvenile hormone epoxide hydrolase gene, and mechanism of action of JHEH from other lepidopterans and vertebrates, and possibilities for considering JHEH as insect biocontrol. In chapter 3, the aims and objectives of the thesis are present. In chapter 4, the materials and methods that are adapted to meet the objectives are present in this chapter. To meet the objectives of the study, the methodology is organized into the following steps. Number 1, cloning of JHH gene in Helicoverpa armigera, expression levels of Helicoverpa armigera JHEH gene during the 5th instar stage of the insect, in silico analysis of Helicoverpa armigera gene, armigera JHEH gene, and its comparative functional studies on par with other lepidopterans, protein modeling, structure analysis, and validation of Helicoverpa armigera JHEH protein, novel molecular biocontrol methods through juvenile hormone binding proteins, JHBP, and RNAi technology. In 5th chapter, the results that are obtained by the above methods are thoroughly analyzed and discussed. In chapter 6, the overall summary and conclusion are present in detail. The possible outcomes of the study, along with the available literature in support of the objectives for developing 3rd generation biopesticides using insects' own enzyme, are summarized in this chapter, followed by bibliography, bibliography, and list of publications from the present thesis work. Thank you.